Okay, so welcome to this next video in which we are discussing the phospholipase C gamma family of phospholipase C enzymes. Okay, right, so we now want to discuss the hypothetical three-dimensional structure of phospholipase C gamma enzymes. And we won't know that this is the structure for sure until we have the crystal structure, but we're pretty confident that this is the structure. Okay, so let me draw this out for you. So basically, the whole thing is dominated by the catalytic domain, which is this tin-like barrel domain. So let me draw this here. Okay, so we've got this large barrel structure here, okay, which is formed by two halves. It's formed by the X box, okay, which is here, this one in front, and then the Y box, which is this one behind. Okay, so it's got two halves. Part of it's formed by the X box, which is at the front here, which I'll colour in yellow here. And then the other half that's behind is formed by the Y box. And remember in the primary structure, there is this large portion that is in between the X and the Y boxes, which is known as the XY linker. Okay, now this is going to form the catalytic domain of the enzyme. So we have this cavity down here, okay, and this cavity has within it a calcium arm. So there's always a calcium arm bound within this cavity in all phospholipase C enzymes. And this seems to be important in the catalytic activity of the enzyme. Okay, so this cavity at the center of the tin-like barrel domain here, this is known as the catalytic core of the phospholipase C enzyme. And basically what's going to happen is PIP2 molecules, phosphatidylinositol 4,5-bisphosphate molecules, are going to stick their heads into this catalytic core. So let me have a go at trying to show this. So here is our phosphatidylinositol 4,5-bisphosphate molecule, and it's going a little bit wrong, but never mind. Here it is, sticking its head into this catalytic core of the phospholipase C enzyme. So I'll try and colour it in, maybe that will improve it. Here are the long-chain carboxylic acids in orange. Here is the glycerol molecule in green. Okay. Here is that phosphate group there, one phosphate group there, and another there. Okay, and then we have the inositol ring in the middle there. Okay, well the message is clear, hopefully. The PIP2 molecules stick their heads into this catalytic core, okay, and what then is going to happen is the PIP2 is going to be cut into diacylglycerol and inositol 1,4,5-trisphosphate. Okay, so that's the XY barrel domain then. Now I'm going to draw another picture of this because this is the enzyme actually performing its role, okay, with the PIP2 stuck in there, but I want to firstly draw the inactive enzyme. Now the inactive enzyme is actually in the cytoplasm, so it's nowhere near the cell membrane, so it's nowhere near the PIP2, which is within the cell membrane. Okay, so it's not currently going to have a PIP2 molecule stuck within that catalytic core. Okay, so here we are, once again, with our X portion and our Y portion of this tin-like barrel domain here. Okay, right, now we want to put this in context of the entire polypeptide then. Okay, so let's firstly put on the XY linker, because this is going to play an extremely important role in inhibiting the enzyme prior to it being activated by a receptor tyrosine kinase. Okay, right, so remember this is the linker between the X domain, the end of the X domain here, and the start of the Y box here. Okay, now if we go back to our primary structure here, remember between the X and the Y, what we had was this split plextrin homology domain. Okay, so I'll start by showing this. So here is the first half of our plextrin homology domain here. Okay, and then we've got the second half of our plextrin homology domain here. So half of the plextrin homology domain was in this portion just after the X box of the tin-like barrel domain, and the other half was just before the Y box of the tin-like barrel domain. Okay, so here are the two halves of our plextrin homology domain, making a full plextrin homology domain here. Okay, then sandwiched in between the two halves of the plextrin homology domain, we had two SH2 domains. Okay, so we had firstly the N SH2 domain, which was closer to the amino terminus. Here that is. Okay, so I'll colour this in blue here. Okay, and then we had the C SH2 domain, which was in between this N SH2 domain and then the SH3 domain, which was towards the second portion of the split plextrin homology domain. So here is our SH3 domain, and I'll show that second SH2 domain in a moment. And the reason I'm not showing it yet is because actually the second SH2 domain, the C SH2 domain, actually folds back and is going to sit within this catalytic domain of the inactive enzyme. Okay, so here is our second C SH2 domain, folded back like so. Okay, so I'll colour this in blue rather than trying to write SH2 there. And this is what is inhibiting the enzyme when it is at rest, basically. Okay, so here, this is our C SH2 domain, okay, which is sitting within the catalytic core and keeping this catalytic domain of the phospholipase C gamma enzyme constantly inhibited. Okay, whereas this one over here, this is the N SH2 domain over here. Okay, so let's just make sure that this tallies up with our primary structure. So we had an X domain followed by the first half of our plextrin homology domain, followed by our N SH2, followed by our C SH2, followed by our SH3, followed by our second half of the plextrin homology domain, followed by our Y box. Okay, so we start with our X domain here. We then go on to our first half of our plextrin homology domain, then our N SH2 domain. We will go round to our C SH2 domain, round to our SH3 domain, to our second half of the split plextrin homology domain here, and then onto our Y domain here. Okay, so now what we need to add in is the things that are prior to the first, to the X box and after the Y box. Okay, so we'll start with the things that are prior to the X box. So remember, prior to the X box, we had these 4 EF hand domains, which we're not too sure what these are actually doing, but they don't seem to actually be binding calcium, which is what EF hands usually do. Okay, so here they are in pink, and in phospholipase C enzymes, both phospholipase C gamma 1 and phospholipase C gamma 2, you have 4 EF hand domains. Okay, so that's true of both of those phospholipase C gamma enzymes. So these are the EF hands. There are some phospholipase C enzymes that don't quite have 4 EF hand domains. Some of them only have 2. Okay, but they all have at least some of them. Okay, then prior to the 4 EF hand domains, you then had a plextrin homology domain. Okay, so a full pH domain here. Okay, and then there was the amino terminus. Okay, right, so that was the structure that we had prior to the X box. And then what did we have after the Y box? Well, we had that C2 domain, so let's add that in now. So after the Y box over here, we have that C2 domain here, and then after the C2 domain, we then have the carboxylic acid terminus of the protein. So I'll colour in the C2 domain in, what colour have I not used? Green I'll use. Okay, right, so here then, this is the supposed three-dimensional structure of a phospholipase C gamma enzyme. Okay, and it's very similar to the structure that we have for phospholipase C delta enzymes and also phospholipase C beta enzymes. The key difference is this elaborate XY linker that is substituted in here for the phospholipase C gamma enzymes, which you wouldn't find in a phospholipase C delta enzyme. So this is either gamma 1 or gamma 2. Okay, right. So this is the inactive phospholipase C gamma enzyme. It's not doing anything at the moment, and at the moment it's within the cytoplasm of the cell, so it's sitting within the cytoplasm, and to be activated it needs to be recruited to the membrane, and then it also needs to be phosphorylated by the receptor tyrosine kinase, and we'll see how this is going to activate it. Okay, so that then is our next topic. We want to discuss how we are going to activate these phospholipase C gamma enzymes via receptor tyrosine kinases. Okay, so to do this, let's start by discussing receptor tyrosine kinases. Okay, so RTKs for short. So these are these are receptors which, when their ligand binds to them, generally dimerize. Okay, and then their tyrosine kinase domain becomes active and participates in a process known as autophosphorylation. Okay, so I will show you all of this now. So we'll use as the sort of prototypical receptor tyrosine kinase enzyme, receptor tyrosine kinase, we will use the epidermal growth factor receptor. Okay, which is a receptor clearly for epidermal growth factor. So EGFR is short for epidermal growth factor receptor. So EGF is the epidermal growth factor. Now, many of the receptors for growth factor molecules are receptor tyrosine kinases, and the epidermal growth factor and the epidermal growth factor receptor are kind of like a prototypical example of all of them. Okay, right. So let's draw this then. So we'll have the cell membrane here, okay, and at the moment we'll just represent the cell membrane by a single line rather than by two lines, okay, and then we've got our epidermal growth factor receptor sitting in the cell membrane. Okay, so in red here, this will represent the epidermal growth factor. Okay, right. Oh, sorry, the epidermal growth factor receptor, rather. Okay. So, in addition, we need to show that it has a very important cytoplasmic domain. Okay, so I'll show this here. So, after the membrane spanning portion, you then have a cytoplasmic domain, and within this cytoplasmic domain, you have a special enzymic portion that has the ability to phosphorylate tyrosine residues. Okay, so this is what's known as the tyrosine kinase domain. Now let me just remind you of the structure of tyrosine and how you can phosphorylate it to produce a phosphotyrosine residue. Okay, right, so let's start with the structure of tyrosine then. So the structure of tyrosine, tyrosine is an amino acid, so I'll start by drawing the core amino acid structure. So here's the amino group, here's the alpha carbon with a hydrogen coming off, and here's the carboxylic acid group. Now I'm drawing an amino acid residue, so that means that I'm drawing it as though it's within a polypeptide. So I've drawn the amino group as though it's involved in binding to the carboxylic acid of the previous amino acid. Okay, and I've drawn the carboxylic acid group as though it's involved in binding to the next amino acid along. Okay, now the R group of a tyrosine residue consists of a methylene group like so, and then with a benzene ring coming off that methylene group here, and then an alcohol group coming off the fourth carbon of the benzene ring. Okay, so this is tyrosine. Okay, now what you can do is you can phosphorylate that alcohol group coming off the benzene ring within the tyrosine residue. So let me show you the phosphate group here. Okay, and what you can do is you can attach that phosphate group via a phosphoester link. Okay, so what you'll do is you'll take the hydrogen off the alcohol group once again, you'll take the alcohol off the phosphate group, you'll bind those two together to make water, and then you'll bind the oxygen of the alcohol group to the phosphorus atom of the phosphate group, and this link that you then forge, once again, is a phosphoester link. Okay, and the whole residue now, when you have a tyrosine residue with a phosphate group attached via a phosphoester link, that's known as a phosphotyrosine residue. Okay, now in reality, tyrosine kinase enzymes, which are the enzymes which put phosphate groups onto tyrosine residues, do not bind inorganic phosphate groups like so onto tyrosine residues. Instead, usually they get the phosphate group off an ATP molecule, so they cut the final phosphate group off an ATP molecule and they then stick it onto the tyrosine residue. Okay, but this gets the basic principle across. Okay, so what's going to happen then is that the epidermal growth factor is going to come and bind to the epidermal growth factor receptor here. Okay, so this box that I'm now colouring in blue here, this is going to represent the epidermal growth factor, or EGF. Okay, now when the epidermal growth factor binds to the epidermal growth factor receptor, the epidermal growth factor receptor is going to change conformation, and this is going to allow it to dimerize with another epidermal growth factor receptor. So I'll show a change in conformation by the extension of this process, which we're going to now use to dimerize with another epidermal growth factor receptor. Okay, so you can only dimerize with another epidermal growth factor receptor which has epidermal growth factor bound to it. Okay, so here comes along another epidermal growth factor receptor with another epidermal growth factor molecule bound to it. Okay, so here is our second epidermal growth factor, and here is our second epidermal growth factor receptor in red here. Okay, and it too will have this cytoplasmic domain which contains this domain which has enzymic capacity as a tyrosine kinase enzyme. Okay, so here in purple is that tyrosine kinase domain. Okay, right. Now, once you've got this dimerization here, the next process that's going to occur is a process called autophosphorylation. Okay, and basically this involves the tyrosine kinase of one of these epidermal growth factor receptors, which we'll call number 1. So let's call this epidermal growth factor receptor number 1, and let's call this epidermal growth factor receptor number 2. Okay, and basically what's going to happen in autophosphorylation, and I'm not sure I'm going to be able to fit it in there, but never mind, autophosphorylation, basically the tyrosine kinase domain of epidermal growth factor receptor number 1 is going to phosphorylate tyrosine residues on the intracellular aspect of epidermal growth factor receptor number 2. Okay, so not only in the cytoplasmic domain do you have this domain that is capable of phosphorylating tyrosine residues, but you also have certain tyrosine residues that are meant to be phosphorylated. Okay, and you'll often have many of these, more than 2 potentially. Okay, so what will happen is the tyrosine kinase domain of epidermal growth factor receptor number 1 is going to phosphorylate the tyrosines on this second epidermal growth factor receptor, and then the tyrosine kinase domain of epidermal growth factor receptor number 2 is going to reciprocate and phosphorylate the tyrosines on epidermal growth factor receptor number 1. Let me try and colour this in. So here in turquoise is the phosphorylation performed by the tyrosine kinase of epidermal growth factor receptor number 2, and here in green is the phosphorylation performed by the tyrosine kinase domain of epidermal growth factor receptor number 1. Okay, so the result of this is that these tyrosine residues become phosphorylated. They become phosphotyrosine residues. Now, these phosphotyrosine residues on the cytoplasmic side of an epidermal growth factor receptor, or indeed of any old receptor tyrosine kinase, because it's a very general principle that you get this autophosphorylation, these are capable of activating all sorts of downstream pathways, but one of them is that they're capable of activating phospholipase C gamma enzymes, which we'll see in a moment. Now firstly, before we just see that, let's discuss why this process is called autophosphorylation. Okay, so auto means self, so this literally means self-phosphorylation. Okay, now you might think that's a little bit odd because actually it's epidermal growth factor receptor number 1 phosphorylating epidermal growth factor receptor number 2's tyrosine residues. Okay, so it doesn't seem as though, it's self-phosphorylation. It seems more as though it's cross-phosphorylation, and some people do indeed call this cross-phosphorylation instead of autophosphorylation. However, the reason it's called autophosphorylation is you view the dimer of epidermal growth factor receptors as being one entity in itself, and therefore it is effectively phosphorylating itself. Okay, so that's why it's called autophosphorylation. So, let's now discuss how phospholipase C gamma enzymes are going to be activated by phosphotyrosine residues within the cytoplasmic domain of epidermal growth factor receptors. But we'll do that in the next video.